So now we have come to lesson 3.2 part 3, and we'll look at the extracellular polymeric substances. These are substances that bacteria produce, and not just bacteria, but actually other organisms as well, such as fungi produce. So they're microbial organic polymers, and they are part of the biofilm production. So the gluey polysaccharides and proteins and other compounds that exist as a result of microbial metabolism that are produced to allow for various bacterial and other microbe functions, but also really support plant growth. These extracellular polymeric substances allow bacterial interactions between bacteria themselves and with their environment. And these are in the diagram here that is taken from this paper called Microbial Extracellular Polymeric Substances, Ecological Function and Impact on Soil, which I highly recommend reading. It's another great review to show the superpowers of bacteria in promoting plant growth. But basically in the diagram you can see the different benefits like drought protection, soil aggregation, adhesion of the microbes to the soil particles and to the plant, how these EPSs actually allow symbiosis with plants, how they play a major function in biofilm structure, trap nutrients, reducing pathogenicity and virulence of various other microbes, or actually allow the pathogenicity and virulence as well of certain organisms. They also provide a carbon source to microbes. They are a way by which bacteria and fungi signal messages to one another and to the plant, so they're a medium by which signaling occurs. They allow for genetic exchange, protection from antibiotics, and also production of antibiotics as well, and exudation of those antibiotics. So one of the major roles of extracellular polymeric substances is the binding of soil particles into microaggregates. But for the bacteria, extracellular polymeric substances provide adhesion of the bacterial cells to the soil particles, but also cohesion, so that the bacteria can work together in a matrix that allows them to communicate and be in close proximity to one another. It promotes chemical reactions and biochemical signaling, so communication, antioxidation, nutrient entrapment, carbon storage, protection, for antibiotics to function, for organisms to protect themselves from antibiotics, also to take away heavy metal stress, drought, and predation. So these are all the different benefits that these EPS molecules provide. What's interesting is that the extracellular DNA that ends up in the soil matrix actually has the potential to change particular bacterial genes and therefore expression, because it can be picked up by bacteria and engulfed and incorporated into their own genes. So it actually increases genetic diversity in the bacterial populations as this extracellular DNA is present. It also allows for signaling and adhesion, and this extracellular DNA actually forms a part of the extracellular polymeric substances, providing structure and cohesion. Abiotic stress actually triggers extracellular polymeric substances to be produced by bacteria, and once produced, these EPS molecules bind and entrap heavy metals and metal binding clay minerals, colloids and oxides, and this actually allows for bioremediation. So when we have bacteria that are stressed in the soil through heavy metal presence, they will produce these EPS substances and actually form complexes with the heavy metals and make the heavy metal inert. So these EPS substances bind these heavy metals and don't allow the heavy metals to cause stress or cause damage to bacteria and as a result to plants. So some of the bacteria, for example, Bacillus species, has been shown to produce EPSs that bind various heavy metals and with a particular attraction to lead at 10 times more intensity than other metals. The EPSs of some other bacteria have also been shown to chelate and deal with different heavy metals. And in this particular instance, the Anabina and the Nostoc have a greater affinity for copper instead of lead here. So different bacteria have the capacity to prevent stress by different heavy metals. Also, the EPSs increase porosity, water holding capacity, and aeration and therefore plant root penetration, allowing for greater plant growth. And this is because they are sticky. So the polysaccharides inside of these EPSs are sticky, so they provide microaggregate formation, sticking soil particles together and therefore forming spaces. They also play a huge role in the establishment of nitrogen fixation symbiosis and nodule formation. So when the EPSs are not present in the rhizobia type of bacteria, for example, the symbiosis won't go ahead and nodules won't be formed. So they're actually necessary in order for nitrogen fixing bacteria to form a symbiotic relationship with their plant to instigate nodule formation. They also provide abiotic stress protection. For example, a Pseudomonas strain has been shown to have EPS molecules that hold water several times their weight and provide bacterial protection. When added to sandy soils, provide more water holding capacity. And arid soil cyanobacteria, their EPSs lead to water and nutrient retention. Sodium chloride tolerance isolates decrease plant sodium uptake and osmotic stress. So isolates of bacteria that actually tolerate sodium chloride can reduce plant sodium uptake and therefore osmotic stress, so allow plants to live in saline soils. And some of these bacteria actually love salt, so they actually can be used in soil remediation in soil salinity situations. Some of them actually can withstand extreme cold and live in ice, and some even in ocean vents where the extracellular polymeric substances allow their survival. So this is the extreme way that they are protected by these EPS molecules in extreme conditions. Certain EPSs in certain species of bacteria allow protection versus antimicrobials such as antibiotics and disinfectants. So bacteria use these EPS molecules to protect themselves from various stresses. One of the most important things that extracellular polymeric substances provide to soil is that of forming soil structure and aggregation. So soil structure depends on the formation of aggregates via vegetation, microbes present, soil fauna, cations, clay particles, and organic matter. So we know this, we have been learning this over the last few lessons as well, but this is such an important concept to understand how soil structure plays a vital role for soil biodiversity and therefore for the healthy plant habitat creation. So some key minerals such as carbonates, like calcium carbonate, iron and aluminium hydroxides, and clay minerals play a key function in aggregate formation. And the extracellular polymeric substances particularly like to find particular affinity for these minerals and really add to soil structure and aggregation. Persistent aggregates, which are 2 to 20 micron in size, which you can see on the diagram here, tiny little aggregates where they just basically use these key minerals to bind clay particles and inorganic binding agents such as oxides and humic substances and polysaccharides and metal ions. So this is the basic structure of these persistent microaggregates, these tiny aggregates of soil that build on that structure and become larger. So larger microaggregates are 20 to 250 micrometers, and they are normally formed via plant roots and root hairs and fungal hyphae. So here is a diagram of this larger microaggregate. These larger microaggregates are formed by the persistent microaggregates coming together through the plant's roots, root hairs, and fungal hyphae. And microaggregates glued by polysaccharides and polyuronides form macroaggregates. So aggregates of size greater than 250 micrometers, which are these larger complexes. And this is where plant and microbial interactions all happen within larger aggregates that are formed of smaller aggregates. And the aggregation process actually provides spaces for water, nutrient acquisition, and oxygen presence. So the microbes and plant roots can actually access and live inside of these structures. Stability of aggregates is very important as it determines internal cohesion, pore volume, connectivity, and pore wall hydrophobicity. So when the pore wall is hydrophobic, it doesn't like water. That's what that means. And it's more becomes more stable to not being disintegrated by water. And so EPSs actually provide this benefit to increase the stability of aggregates. Soil aggregation is fundamental to soil productivity and environmental quality because it actually allows water and oxygen to be present within the soil and prevents erosion, and also prevents runoff of different chemicals into waterways, allowing soil to detoxify whatever is present that isn't good for life. So it is very important to understand that soil bacteria produce these EPS molecules and how these molecules play this absolute necessary structural component of ensuring soil structure and soil aggregation. So further on to that, bacteria and fungi increase soil structure via these extracellular polymers and also through degradation of aromatic humic materials. And that leads to clay metal organic matter complexes. So that's another way that soil is aggregated. Extracellular polymeric substances also lead to organomineral associations, giving rise to immobile and mobile organic matter composition and also mineral reactivity. They adsorb to mineral surfaces, providing connectivity and water retention, so therefore aggregation. And of course, the EPS molecules all depend on the species of bacteria and the soil substrates and management, the substrates present in the soil that feed and allow these bacteria to grow on, and how we manage the soil ecosystem in order for the EPS molecules to function properly. So some of the bacterial species, such as Bacillus and Pseudomonas, are known to stabilize and increase aggregate strength, and some of them have specific ways in which they do that and are more effective than others. It has been shown that in irrigated and no tillage conservation soils, there are increased aggregating bacteria populations. Pseudomonas putida has been shown especially to produce large amounts of EPS and therefore increase aggregate stability in over 50% of soils that are affected by temperature, salt, and drought. Bacillus has been shown to be a strong EPS producer, and over a period of 1 year in desert experiments designed to increase soil structure, this bacterium has been shown to increase heterotrophic community of other bacteria, as well as bacterial numbers and nitrogen and phosphorus. So through the production of the EPS, Bacillus has been demonstrated to strengthen the bacterial community and nutrient cycling. The specific EPSs that it produces are glue-like polymers that connect sand grains together. And these type of bacteria can be used in soil structure recovery under nutrient and water limitation. So basically, knowing which of these bacteria prefer specific soils, we can then target the production of these bacteria in those soils in order to generate remediation of these soils or restoring these soils so they can promote plant growth. And of course, I'll always say that the biodiversity of these bacteria is essential. So not only numbers and specific species that have been studied, but rich biodiverse communities must be ensured through the delivery of these communities into the system and ensuring their survival through specific management practices of these soils. In desert ecosystems, like you can see here on the left, cryptogamic or biotic crust is where there is an interwoven community of cyanobacteria. So these are bacteria that can actually photosynthesize. Green algae, which are also photosynthetic, and microfungi, bacteria, lichens, and mosses work together as a community of various organisms, creating these structures and making sure that soil is actually held in place, that it doesn't just blown off and keeps getting lost. And you can think of these cryptogamic or biotic crusts or cryptobiotic crusts as little ecosystems that form that allow plant seeds to germinate, and they're like the beginning process of soil restoration, where all these organisms come together to prevent further damage to the soil and to encourage biodiversity in the soil, to encourage growth and restoration of the soil ecosystem. Cyanobacteria play a crucial role in soil restoration, and they are photosynthetic organisms. So that means not only do they provide the EPS forming capabilities to restore soils, but also provide carbon in the sense that they can fix carbon because they photosynthesize and therefore allow that first line of organic matter to be developed. They play a key role in cryptogamic crusts, especially in arid ecosystems, leading to soil fertility, prevention of erosion, and decreasing soil temperature, carbon and nitrogen content accumulation in the soil, aiding in the hydrology and maintenance of good water balance, and therefore supporting seed germination in damaged soils or in arid ecosystems. They increase soil structure, fertility, and nutrient bioavailability, and markedly increase quality of arable lands, but also degraded and desertified environments. Cyanobacteria can be viewed as stress-tolerant, drought-resistant oligotrophs, and they have very specific extracellular polymeric substances that they produce, creating a gluing mesh that increases soil stability, moisture, and biological activity. Their EPS is also hygroscopic, which means that it can absorb moisture from the air. The cyanobacteria and the EPSs produce hard, entangled superficial structures, providing for stability of semi-arid soil surfaces and erosion protection as a result. Nostoc muscorum, which is a species of cyanobacteria, increases aggregate stability and soil aggregation by 18%, as well as carbon by 60% and nitrogen by over 100%. It also increases microbial populations and increases lettuce seedling emergence by more than 52%. Combination of variety of organisms and functional groups has far greater potential than the actual individual species. Once again, we're coming down to biodiversity. We need biodiversity of microbes. We can't just be looking at individual taxa or individual species and saying we've found the miracle cure for our soils. This is all about creating communities, microbial communities that are self-assembled, as you can see from the cryptogamic crust example. They just need the conditions in order to do that, to proliferate, and that means no further soil disturbance in order for them to function as a community, performing their work in restoring soil ecosystems. So what happens when we actually inoculate these EPS producing bacteria onto plants? They definitely show protection of the rhizosphere from drying and moisture fluctuations, increased root exudates and nutrient uptake, as well as therefore plant growth. Obviously, as we've already mentioned, the EPS also increases soil aggregation and structure, as well as nutrient and water uptake and seedling growth. Rhizobium isolated from an arid soil has been shown to actually be a weed growth promoter. It's a prolific EPS producer that increases soil structure, root adhering soil, which you can see here on the left. It's basically a rhizosheath. So the EPS allows the soil particles to be glued to roots and therefore provide habitat for microbes and all the processes that are required for plant growth to be enhanced. It also increases aggregate stability and reduces, as a result, soil water stress. Sunflower plants that have EPS producing bacteria, or EPS actually added directly to the soil as well, have shown an increase in soil aggregation, water and nutrient uptake, formation of a root adhering soil, and soil micropore volume, but also growth, shoot, and root biomass is increased in these sunflower plants. Also, other experiments have shown yet other bacteria that have been added to the wheat rhizosphere, producing significant aggregation, stabilization, RAS, and increased water stable aggregate diameter. So these are just examples of what is possible when these EPS producing bacteria are actually inoculated onto plants. In this particular case, it regulates water content in the rhizosphere, So we know that the EPSs actually regulate water content in the rhizosphere. And also on chickpeas under elevated salinity, when these chickpea plants have been inoculated with two different species of EPS forming bacteria, there has been an increase in plant growth and aggregation by over 75%. So lots of benefits, lots of fabulous benefits to the plants, because bacteria are just these really cool organisms that have fantastic capabilities that we are just learning about and beginning to discover. So there's lots more to be learned in this as research progresses. And as always, I will say, let's create soil biodiversity and promote that biodiversity, because we know the science is there. We just need to create the conditions for these organisms to thrive. So we have now come to the end of lesson 3.2. And so the summary of that is that we have looked at plant growth promoting rhizobacteria. We have covered the amazing process of rhizophagy. And just now we finished looking at extracellular polymeric substances that are produced by bacteria and how this helps soil structure and plant growth. So I will leave you with these references. These are all the different amazing references that I have used, and I encourage you to go into these and read for yourself about the incredible magical powers that bacteria provide and how essential they are to plant growth and therefore to our own existence. So I'll see you in lesson 4, which is on fungi, and wishing you all the best. Bye for now.